Welcome to Vivid Botany. We are once again looking here one question from molecular basis of inheritance. This chapter is very crucial. It's a uh, very key to understand the entire botany, biology. Let us see the questions, the statements one after the other. In capping, methyl guanosine triphosphate is added to third end of hnRNA. Let us see first hnRNA means what? hnRNA is heterogeneous nuclear RNA. hnRNA stands for heterogeneous nuclear RNA. This RNA is precursor of mRNA. That is, mRNA is formed from this hnRNA, and this hnRNA is formed from RNA polymerase 2. RNA polymerase 2. There are three types of RNA polymerases: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. RNA polymerase 1 will give rise to ribosomal RNA, that is 28S, 18S, 5.8S. RNA polymerase 2 will give rise to the mRNA, first hnRNA. hnRNA undergoes certain modifications and that converts into the mRNA. RNA polymerase 3 give rise to tRNA, or we call it as transfer RNA, 5S rRNA. That is small nuclear 5S rRNA, another is snRNA. That is small nuclear RNA. Right. Now let us see, this hnRNA contains, that is, in eukaryotes, hnRNA contains both introns and exons. It contains introns and exons. What are introns and what are exons? Simply remember that introns are not useful at that time, but exons are useful at that time. So what happens here? These introns are removed. Removal of introns is called as splicing. This splicing is done by splasiosome, that is snRNA, small nuclear RNA that is produced from RNA polymerase 3, and having exons. What are exons? The functional genes. The useful genes at that time are called as exons. So these exons are retained. So removed are introns, retained are exons. These removal of introns, rearrangement of exons is done in hnRNA. Then hnRNA will convert into messenger RNA, right? And removal of introns is called as splicing, which will cause splicing snRNA, that is small nuclear RNA. And having introns and exons is called as split gene arrangement. Split gene arrangement. This is having introns and exons is called split gene arrangement, which is not seen in prokaryotes. That is, in prokaryotes there is no necessity of splicing as there are no introns in prokaryotes. So the processing of mRNA is not like that in prokaryotes. Directly, once transcription takes place, that mRNA can undergo translation itself the same time. That is, on one end transcription will be going on, the other end translation will be going on, as there is no splicing, as there is no processing of mRNA. And moreover, do remember that in prokaryote there is no separate chamber for nucleus. So transcription, translation both will take place in same compartment in prokaryotes, as there is no compartmentalization in prokaryotes. So we understand that there is no split gene arrangement in prokaryotes, there is no splicing in prokaryotes, and transcription, translation both will take place same simultaneously in prokaryotes, and in one compartment transcription, translation both will take place in the prokaryotes. Right. Now let us see the second statement. By the way, what is capping here? See, I told you that After removal of the introns, the exons are rearranged. So this will be that mRNA, precursor of mRNA. Precursor means from this mRNA, the complete processed mRNA is formed. Let us suppose this is fifth end, this is third end. Now what happens? At fifth end of this mRNA, guanosine, methyl guanosine triphosphate is added. So this is guanosine, this is methyl, this is triphosphate is added at fifth end of hnRNA. This addition of methyl guanosine triphosphate is called as capping. So we should know that the capping should be done at fifth end of the hnRNA, and tailing should be done. Tailing means poly A tail. Poly A tail. Poly A tail means about 200 to 300 adenylate residues. Adenylate residues are added at third end of the mRNA. So this process is called as tailing. Remember, the capping, splicing, tailing, all that takes place only in eukaryotes and not in the prokaryotes. Now let us see the first statement here. In capping, methyl guanosine triphosphate is added at third end of hnRNA. No, it is not correct because it is not third end but it is fifth end. In my previous videos also I have told you that DNA replication, transcription, translation, movement of ribosome on mRNA takes place only from 5 to 3 but not from 3 to 5. Do remember, at fifth end the capping is done, at third end the tailing is done. 5 to 3, so 5 is face, 3 is tail. 5 is cap, the 3 is tail. Do remember in such fashion. Second statement, RNA polymerase binds with Rho factor to terminate the process of transcription in bacteria. See, RNA polymerase binds with the Rho factor. Remember always, RNA polymerase is meant for polymerizing the nucleoside triphosphates to form that mRNA. So here two factors are there. One is sigma factor, another is the Rho factor. Sigma factor is called as initiator factor, Rho factor is called as the terminating factor. I write here, this is sigma factor. It is meant for initiation, and this is the Rho factor, RHO. It is meant for termination. So sigma factor is initiation factor, Rho factor is termination factor. He is saying that RNA polymerase binds with Rho factor to terminate the process of transcription in bacteria. So this is very much correct. It is the answer here. Let us see third one. The coding strand in transcription unit is copied into an mRNA. Right. This is wrong here. How do we understand this? See, always remember that an mRNA is formed from DNA. Let us suppose this is the DNA. This is 5 to 3 and this is 3 to 5. Now, in the two DNAs, one DNA will give rise to mRNA and that DNA is called as template. Let us suppose here this DNA is giving rise to the mRNA. So this acts as template. The other DNA strand which doesn't give rise to the mRNA is called as coding strand. So this is template strand. How do we understand this? From the template strand, the mRNA is formed like this. So this is mRNA formed from template strand. That's also from 5 to 3 direction by the enzyme RNA polymerase. We call it a DNA dependent RNA polymerase. So this mRNA is complementary to template strand. Let us suppose here C is there. In mRNA we have G. Here G is there on template. Here C. Here A is there. Here there is no thymine because RNA lacks thymine. It contains here uracil. Here thymine is there. Here adenine is there. Here adenine is there. Here uracil is there. Cytosine. Then guanine. So mRNA, that is formed from template is exactly complementary to that of template. And coding strand, if you see here, this is guanine, cytosine, and this is thymine, adenine, and again thymine, again guanine. So if you see the coding strand and this mRNA both will be same. So that this is called as coding strand, which is not giving rise to mRNA. Except in the case of thymine, everywhere coding strand and the mRNA both will be same. And mRNA is complementary to template strand and mRNA is identical to the coding strand. Now let us see the third statement here. The coding strand in a transcription unit is copied into mRNA. Is wrong. Template strand is copied into mRNA during transcription, but not the coding strand. You have understood, I think. Template strand is copied. Template strand is copied into that, what do you call, mRNA, I mean complementary, and coding strand is not copied into the mRNA. The split gene arrangement is characteristic of prokaryotes. Split gene arrangement is characteristic. I told you that split gene arrangement present only in eukaryotes. It's not present in prokaryotes. There are no introns in prokaryotes. So when there are no introns, there is no splicing, there is no capping, there is no tailing, there is no processing of mRNA. All that are not seen in the prokaryotes. So this statement is also wrong. So this is really very beautiful question framed here. Simplest statements are given. One can understand them very easily. But you should have the basic knowledge of what is RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, what is template strand, what is coding strand, how the messenger RNA, hnRNA is formed, what is capping, what is tailing, what is splicing, what is snRNA. All this you should have a good knowledge, and what is sigma factor, what is rho factor. Sigma factor is asked last time. Last time this question is asked, I think in 2019, sigma factor is asked 2 years back. And this time also they will ask the same questions on these concepts. So be ready. I request you to draw the diagram from the page of NCERT and try to know that what is exactly the sigma factor, what symbols also. The sigma factor symbol, rho factor symbol also you should be very perfect, and where capping is done, where the tailing is done, what is snRNA, right? What is this termination factor, what is template strand, why it is called template strand, also called as antisense strand because it is complementary to the sense strand, and coding strand also called as the sense strand. So this mRNA is formed from template strand but not formed from the coding strand. So I think my video is helpful for you to concretize your concept, to boost your confidence, and to make the next NEET very perfectly without any difficulty. So not to forget to subscribe my channel. We are discussing all this in Vivid Botany. I will be explaining you the rest of the questions of 2021 also, one after the other. Hope we are doing well in the discussion. Why I am telling this much detail every question means, it doesn't mean that only to explain this question, but I want to explain all the terms which are given in the form of statements or the form of maths are following, so that if any question is framed on the other terms also, you can do that very well. Just to clarify your concepts in a very neatly, I am doing all this. Otherwise, just I could have told you that this is wrong, this is right, this is wrong, this is right. I can do like that, but it's not my intention to that. See, I am not a fast going person. I want to make it as slow as I can. At the same time, I want to clarify your doubts. I want to concretize. I want to make you very much perfect. Because once you read very perfectly, again you need not read that. Again you need not go to that page. Again you won't get any doubt from that page. That is my intention. What I say that though you are very slow, though you are a slow reader, need not worry. But when you are going that page, please make note each and every keyword, examples, diagrams, labelings very perfectly, and see how a question can be framed. How many types of questions can be framed from that para, from that illustration. That is more important while you are reading the page. And my intention also not to just rush over the questions, but to explain them as much as I can so that children will not get any doubt further and they can answer any type of question that is asked based on this concept. So let me stop here. We will see next question. Thank you.